So it's my really great pleasure to introduce Jeff Buki to you today. He bailed out of a DARPA meeting in Austin yesterday to come up here and actually make this presentation for us. So I think it reflects a real commitment to try to help our community understand some of the opportunities that are in front of us and some of the things that we might be looking to get into in the next decade. Jeff is the founding director of the Institute for Systems Genetics at New York University Langone Medical Center in New York City. And he works on writing genomes and primarily in yeast. And the two of us met when we were at a scientific advisory board meeting for Recombinetics recently up in Tahoe. And I saw him give this presentation and it just blew me away. So I thought, this is the guy that we need to come and have tell us about writing genomes, and I think you'll be really excited by his presentation. So with that, thank you very much for coming.
All right, well, thank you very much for having me. This job that I have introduces me to a lot of different communities, and it's really exciting to be able to tell you about what I think is in all of our futures, which is genome writing. So before I start, I just want to acknowledge my conflicts. I am involved with not only Recombinetics, but several other companies that are interested in using genome writing and synthetic biology in general to solve a variety of complicated problems. My institution, NYU, requires me to use their logo on all of my slides, but being a bit of a rebel, I insist on using a version of it that's written entirely in engineered yeast. Those are engineered yeast colonies that produce pigments of different colors, and I'll talk a little bit more about that at the end. So what we've been doing for the last 10 years in my lab is working on the next generation of Saccharomyces cerevisiae, that wonderful organism that produces beer, bread, wine, and a multitude of other industrial products today. It is the darling of the fermentation industry, so it has tremendous practical importance as well. It's the first domesticated microorganism domesticated by our species. And what we, colleagues of mine at Hopkins, where I spent 28 years before moving to NYU, decided about 10 or so years ago, Dr. Chandra and Dr. Bader and I, was to dream up what we would do if we could write an entire genome from scratch. And we call this SC20 for Saccharomyces cerevisiae 2.0. And you can't see it on the background here, but there's kind of a blueprint in the background of SC2.0. But what I've outlined here are some systematic changes that we designed into this genome on a computer and then basically built it. So in my previous life, I worked on mobile DNAs, transposable elements that move around in the genome, repetitive DNAs that certainly in the yeast, we have reason to believe we can obliterate, we can exterminate from the genome by essentially editing them out in a computer. There are many other types of repeats, like subtelomeric repeats. We also decided that with Saccharomyces, we could probably remove all of the introns, because only 5% of yeast genes have introns, and they typically have only one, very different from a mammalian structure. And there was reason to believe that we would be able to do that from other people's work. We also moved some genes around. We also decided to relocate some genes, namely the transfer RNA genes. There's 275 of them. And we thought that the genome looked messy, with tRNA genes sort of higgledy-piggledy scattered throughout the genome. And so in an effort to re-engineer this genome, we thought it would be nice, since they're all transcribed by one type of polymerase, to give them their own special chromosome. And there was another reason that we did this, which was that tRNA genes are known to be, to lead to genome instability. When replication forks and the specialized polymerase that transcribes them collide, you can actually have a DNA break that then leads to genome rearrangements. So the theme here for this delete and relocate primarily is to try to build a genome with enhanced genome stability by removal of repeats and so on. Now, in a different mode, we also added a system that we call the scrambling system. Basically involves dropping LOXP sites, or site-specific recombination sites, precisely in the 3' untranslated region of all of the genes. And this would then enable us to scramble the genome and produce many, many variant genomes. So this is a kind of inducible genetic flexibility that we can try to harness to learn more about the genome. And then finally, we do a recoding of small snippets of DNA that we can use to watermark the sequence so that we can distinguish the synthetic DNA we made from the native DNA that's in the genome. And we also took a page from George Church's book and converted all TAG stop codons to TAA stop codons. You may be aware that there's been a whole industry for the last 20 years of taking specialized systems called orthogonal tRNA synthetase pairs that recognize the TAG codon and can insert a custom amino acid of your choice. And we thought that this freeing up one of the 64 codons would be an interesting way to engineer the genetic code and introduce additional amino acids into it. This could be used also for biosecurity, for making the cell dependent on an exotic amino acid not found in nature, and of course open up all kinds of possibilities in terms of new protein chemistries. But fundamentally, SC2.0 is a designer, fully synthetic genome that is synthesized from oligos. So every base pair at the end of the day comes from oligos. So everybody wants to know, well, how the heck do you do that? And this is a very high-level view of how we do that. We basically start with oligos. We build them up into ever larger DNA molecules. And the size of DNA that goes into the yeast we call a megachunk, okay? It's a 30,000 to 100,000 base pair piece of DNA that we assemble in vitro from smaller parts. And it consists of a synthetic segment, a genetic marker, and a tag here of endogenous sequence. So here's a resident chromosome in the living yeast cell. It has another selectable marker here. And basically, the first megachunk in has a synthetic telomere at the left end, so a seed sequence that will grow a new telomere when it goes into the yeast cell. And at the right end, it can undergo recombination with the endogenous chromosome. We can select for this marker, and we can screen very easily for the loss of this marker. So we should get blue and then cross over into the black. And we get that at a very high frequency. And the subsequent megachunks are designed to recombine at both ends. So we get a double crossover. So now we get blue, green. We overwrite the blue marker and we bring in the red marker. And these moves are very easy because it's a microorganism. And so we can just iterate this and go back and forth between the two markers. We call this swap-in for switching auxotrophies progressively for integration, but also to indicate that we're swapping in one synthetic segment at a time and essentially testing how well does it work, does the yeast grow well, and so on. And so after 10 or 11 steps of this, you get this multicolored chromosome, so to speak, that's entirely reflecting the design that was first done on the computer. So that's how we do it, and of course we can have, there are 16 chromosomes, so we can have 16 teams synthesizing the 16 chromosomes in parallel. But before we do any of that, we have to actually design the sequence, and this is where my colleague Joel Bader came in. I don't know if it's possible to dim the lights a bit, because it's a little hard to see, but what you're going to see is a movie here of chromosome 5, and how it's been changed to, there we go, how it's been changed on the path to SC2.0. So you see the genes have become watermarked with those gray bars, and they're jumping around because the designer is moving from left to right and deleting segments and so on. You'll also see these green diamonds, which are the LOXP sites that we introduced at the 3' end, and you'll see some pink dots indicating where we've swapped one of the stop codons and so on. So this amazing software was developed by Joel Bader and his colleagues at Hopkins, and this chromosome now exists, and it was built by basically a team of undergraduates at Tianjin University in the Build a Genome China class. They did all the synthesis and assembly, every base pair of it. And it's an extension of a course we started at Hopkins called Build a Genome a few years ago. So we realized relatively early in the game that this was a huge project. It was not going to be a one lab project, and it would be a lot more fun and interesting if we could recruit a big team, a global team, to do this together. And this was supported by the NSF, and here you can see the US teams and the 3 Chinese teams, Australian teams, Singapore, and UK teams that collectively cover all 16 chromosomes, plus the tRNA NeoChromosome, the specialized chromosome for the tRNA genes that we have put together. This is another view of the international project where you can see that all of the chromosomes, of course, you know, God did not create them all to be the same length, right? Some of them are short and some of them are long, and guess which ones we picked to start the project? Well, of course, we started with the small ones, and so you'll see a lot of green here among the small ones because the green is the color we chose for the US on this slide. And then we started like trying to convince our colleagues overseas to do this, and they said, wait a minute, you're giving us a chromosome that's twice as big as anything you've ever made. And we said, yeah, but we know you can do it. And then we felt very guilty, so we took on chromosome 4, which is the largest chromosome in the genome, and we're now making great headway on that. So you can see also we have an amazing consortium of different types of organizations involved in this, including government labs, private industry, universities, of course, research institutes, genomics giants like BGI, and my favorite, the Australian Wine Research Institute, which brings the refreshments to our annual meeting. So we've designed all 16 chromosomes using BioStudio, and we have recently published in March 5 papers, actually 7 papers, but there are now 6 completed chromosomes, start to finish, sequenced, debugged, and validated. And these are all living yeast cells that contain 15 native chromosomes and 1 synthetic chromosome, and they're high fitness, meaning they behave just like normal yeast, except we've endowed them with special property that their genome can be scrambled and so on. So that's the status. I can tell you that the vast majority of the rest of the genome is all synthesized and assembled and in various stages of finalization. So we think we will be completely finished with the assembly and debugging phase by the end of 2018. And then what will remain is to put all 16 chromosomes together, but we've developed a good way to do that as well. So I'll just tell you one story about one of my favorite chromosomes. This is chromosome 12. And the synthetic version of it is SYN12. And one reason that it's particularly interesting is it houses the ribosomal DNA. And the ribosomal DNA is encoded by a series of repeats totaling about 1.5 megabases in length. So about 150 repeats of a 10 kb unit that encodes the major ribosomal RNAs. And this project was done by Junbiao Dai in Tsinghua University, a former postdoc and a very great member of our consortium. And this is the biggest chromosome that's been completed to date. It's a megabase. And to break up this job into smaller parts, he basically broke it down into 6 subsegments and had 6 teams going within his group. You can only do this in China. And each one making 1/6. And then using meiotic recombination to combine these into ever more synthetic versions, and ultimately 100%. So he succeeded. And then the question was, what about the rDNA? So he started the project with just leaving the native rDNA there. Earlier work by Nomura in UC Irvine had shown that it was possible to build a yeast that didn't have an rDNA locus, but was kept alive by a plasmid-borne copy of ribosomal DNA. The cell doesn't like it, but it survives. And then you can go back in and reinsert the ribosomal DNA, and the cell is happy again. So he thought that this would be a good thing to try, because in this manner we could, in principle, engineer the ribosomal DNA itself and make it different from the wild-type synthetic version. So anyway, I'm not going to go through this because it's all published old technology developed mostly by Nomura, but suffice it to say there's a way to do this. And one of the things that he decided to do is not only to put the rDNA back into its native location, which is very far from the centromere on chromosome 12, on one of the longest chromosome arms in the genome, but he also put it in some other locations, including one on a small chromosome, chromosome 3, that was quite close to the centromere, and you'll see why that's important in a moment. So, because we knew this was going on, and because we knew about the revolution in genomics called high C and all of its various C's, we were very interested in the question, how does the trajectory of the chromosome through the nucleus change when we make all these changes to the chromosomal DNA? So you may be familiar with these contact maps. Sorry, the resolution isn't coming through perfectly, but the contact maps basically consist of a strong diagonal. So we have all 16 yeast chromosomes on the X and Y axes here. And the strong interaction of the primary sequence, things that are close together in the primary sequence, is reflecting here. What you can't see in here, but I can see it on my screen, is that there are actually 16 off-diagonal dots on each chromosome, and those are the 16 centromeres, because the 16 centromeres are held together by the spindle pole body, and that's reflected in this contact map. So one question that came up, and I'm sorry for the resolution here, but was, well, how does just a random chromosome, forget about the rDNA for a minute, how does that compare? And here we have the synthetic version of chromosome 2 and the wild-type version of chromosome 2. Now, what people who study high C tend not to show you is the raw data. But this is the raw data, okay? Then you get these white stripes because there are big repeated regions on the chromosome, big transposons, and most people who display these things just filter that out. But we didn't have to filter anything out of SYN2 because we've removed all the repeats from the chromosome. So you actually get a much nicer, higher resolution picture from these synthetic chromosomes than you get from native chromosomes. And here you can see, for example, the centromere, there's a pericentromeric region where there's a lot more off-diagonals. And you can see that there's a great degree of symmetry in the pattern, forgetting about the white stripes. So what that tells us is that when we've done this now for comparisons of all the synthetic chromosomes we've done, is that the trajectory that the chromosome takes through the nucleus, so here's SYN5 versus chromosome 5, is very similar, despite the fact we've taken all the tRNA genes off. They're supposed to be really important for chromosome cohesion and condensation. They're all gone, but the chromosome trajectory looks the same. The repeats are gone. The subtelomeres are gone. So what that says to me is that those things don't have anything to do with the trajectory of the chromosome. One thing that matters is where the centromere is. All the centromeres are clustered up here at 12 o'clock because that's where the spindle pole body is. And the nucleolus is down here at around 6 o'clock in the images I'll be showing you. Now we can even do this with more than one synthetic chromosome. So here's one that has SYN5 and SYN10. And again, you see that the trajectories of the chromosome through the nucleus, this would be similar to chromosome territories in mammalian cells, are largely unaffected by these extensive modifications that we've made. Okay, so back to chromosome 12. Chromosome 12, I kind of lied to you when I said that the diagonal, the chromosome interacts only with itself and not with the other chromosomes. In the chromosome 12, you actually get this checkerboard type pattern, and the position in the middle here is the ribosomal DNA, because the DNA to the left and right of the ribosomal DNA basically is not interacting. So it's like these are two separate chromosomes. When we remove the rDNA from SYN12, the checkerboard goes away and it behaves like all the other chromosomes. And here what you can see is chromosome 3 looks like any other chromosome. It has a strong diagonal. It doesn't interact with the other chromosomes. But when we transplant the rDNA in there, it picks up the checkerboard because it's now got this huge ribosomal DNA in there. Now the dotted circles here represent the inferred position of the nucleus, where all the ribosomal DNA is. And what you can see is in the native and synthetic version of chromosome 12, the nucleolus is approximately in the same position, and the trajectory of the chromosome is approximately equivalent. But when we transplant the rDNA onto a small chromosome, here, chromosome 3, it's now been pulled up by the, by the centromere cluster. Okay, sorry about that. But there also seems to be some force pulling it southward because there's something that's forcing the nucleolus away from the centromere. Now, to accommodate this giant blob of ribosomal DNA and ribosome assembly that happens in the nucleolus, all of the other chromosomes had to shift in position. So there's been a massive change in trajectory of all of the other chromosomes to accommodate this shift. And the amazing thing is that this cell grows just fine. It has a normal, relatively normal transcriptome. So one of the things that we're learning from this is that we can make massive changes to genome structure with only minimal impact on fitness, which is very surprising. Okay, the second thing that we can do is we can do this genome scrambling. So what we have a complicated acronym here that's not worth going into, but basically we've inserted LOXP sites throughout the genome. LOXP is a 34 base pair sequence that's recognized by the Cre recombinase, of which we have a chemically regulated version, so we can introduce this chemically regulated Cre at any time after we synthesize the chromosome. We can flip a chemical switch and trigger a formation of a huge population of genome variants, or scramblants as we call them. And those will have deletions, inversions, translocations, and duplications resulting from interactions and recombinations that occur between pairs of LOXP sites. So the basic idea here is we start with a uniform population of yeast with a synthetic chromosome or multiple synthetic chromosomes. We flip on Cre and we get out a huge set of variants, and some of them will be lethal, but it's easy to study the survivors, and so we've done quite a bit of that now. And the idea is that we can use this to also develop strains that do better under a particular condition. So, for example, for industrial biotechnology, you might want to select for something that makes more of a useful product. So another way to think about this is it's a way to shuffle the genes in the genome, so this is kind of the elevator pitch for Scramble. If the genome of yeast is 5,000 genes, then think of that as a deck of 5,000 cards. So with scrambling, we can shuffle the deck just like you would normally shuffle a deck of cards, change the order of the genes, but we can also cheat at cards because we can remove any card we don't like from the deck, and we can even put 10 jokers in the deck if we want to. Okay, so it really opens up all kinds of doors in genomics. So some favorite topics: how many of the genes can we remove and still have a living yeast? What are the trajectories to minimal genomes? And can we get gain of function by adding extra copies of genes? How much scrambling of a genome would lead to reproductive isolation and potentially make a new species? And can we rewire gene networks using this scrambling? And if we scramble a genome, can we find a yeast that makes more of a useful product for biotechnology? So these are all things we're interested in. Obviously, I'm not going to tell you about all of those things, but I will tell you that we've scrambled this molecule. It is a 90 kilobase, 100 kilobase plasmid, basically. It represents one chromosome arm. Happens to be in the form of a circle. This was the first one that we made. It has 43 LOX-P sites, and thus 43 segments that can be scrambled. And this was our first collaboration with BGI. And what we did was we created this Scramblegram, which is a visualization of a whole bunch of variants down here of a parental structure. So I'm going to zoom in because I know that's hard to see. But there are 43 triangles pointing to the right here and colored by the colors of the rainbow. And here you see that some of the triangles have flipped around. Here's some yellow triangles that have moved from up here to down here. Massive deletions in this one, massive expansions in this one. These are all high fitness variants. Okay? So they go from a 40% reduction in genome content to a 3.7-fold amplification in genome content for this 100 kb region. And importantly, none of the variations are the same. They're all different from each other. So it really does work to generate tremendous genome diversity. So anyway, our team is using this for all kinds of purposes, but I just, in the interest of time, I want to cover two more topics that I think are of interest to your community. One is what we call NeoChromosomes. So everything I've talked about so far is rewriting the base genome of yeast, which at some level is of absolutely no interest to you. But it might be interesting to add a custom chromosome to perform a custom job. That's something that should be interesting to everyone. We can imagine, well, in fact, we know that there are some cows in this country that carry a bovine artificial chromosome that confers a special property on them. So, so we've been putting a lot of effort into this in yeast, and obviously we're transitioning this to higher organisms as well. But basically, what we call a NeoChromosome is, it has the parts of a chromosome: centromere, origin, telomeres, and then it has some pathways, and it can contain some essential genes that lock it in to the genome of its host, because those genes are essential and have been deleted from the host. So there's lots of things you can do with NeoChromosomes in yeast. That one of them is make the pretty colors that I showed on the first slide. So this yeast is engineered to produce a purple pigment by expressing 5 genes from the bacterium Chromobacterium violaceum. And these are genes that produce carotenoids, healthful compounds produced by a variety of other microorganisms. And it's hard to see, but we have orange and yellow and purple here. And so these are very simple toy systems that we use to optimize the construction of NeoChromosomes. Here you can see how we've sort of shuffled around different promoters and terminators and gotten very different levels of color by scrambling these systems in a combinatorial way. Another thing that we can do is transplant an entire pathway from one organism to another. So one of our first efforts in this has to do with transplanting a metabolic pathway, namely the Denovo Pathway to the synthesis of adenine. So this would be the yeast genes in red and the mammalian genes in blue. These are very highly conserved, so it's very easy to identify the orthologs. And basically what we do is build all of those human genes into a NeoChromosome, and then we can knock out all of the yeast genes and see whether or not it works. So our basic strategy here is we take the coding sequence from the donor organism, the promoter and terminators for the corresponding genes from the yeast, and then we string those together into a NeoChromosome, and then we can build what we call a pathway transplant strain, where we've knocked out all of the yeast genes and we've brought in all of the human genes. And what you can see is the strain with no NeoChromosome cannot grow in the absence of adenine. When we give it the NeoChromosome, it grows. So it works. It basically worked the first time, but interestingly, it didn't work perfectly. This is how the wild type grows, nice big colonies, and this is how the NeoChromosome strain grows, little tiny colonies which eventually grow up. And since we did this in a stepwise fashion, the deletion of the host genes, we could see where the doubling time shifted from normal to much longer than normal. And it had to do with deletion of the first gene in the pathway, ADA4. And so the human counterpart, PPAT, doesn't fully complement the ADA4 mutation and leads to the longer doubling time. So anyway, I'm going to skip over this in the interest of time, but the interesting thing here is this is not an expression problem. We can overexpress the human PPAT as much as we want. It never complements the ADA4. So there's something fundamentally different at the protein level. And so one thing we've done with our expanded synthesis capabilities, this is kind of trivial, is we've now looked at the PPAT, or ADA4, from 70 different organisms. And it turns out that the ability to complement clusters phylogenetically. So all the mammals, almost all of them, complement exactly like human. They complement, but not very well. Here we have a group of organisms that don't complement at all. The microbial ones all complement well, except for archaea. Archaea doesn't work at all. So what we conclude from this phylogenetic clustering is that the fundamental metabolic state of these different organisms is different, and ADA4, being a critical regulatory step in the pathway, is very sensitive to some difference in metabolic state between these different organisms, and it's tuned. So, what we think is, because this is a highly regulated allosteric protein controlling the rate-limiting step, it basically is telling us that orthologs from species with similar lifestyles maybe have similar metabolism. And anyway, that's probably the most interesting conclusion we have from this particular study. We've also transplanted human nucleosomes into yeast. Okay, so imagine that the core nucleosome that we package our DNA in is consists of the 4 core histones. We've been able to delete those from the yeast and substitute the 4 major human histones for it. We get an extremely sick strain. It has to accumulate a series of suppressor mutations to survive. And if you're interested in it, I'll be happy to give you a lot more information about it. We have a paper coming out in the middle of December on this, and it's on bioRxiv if you're interested in this. But some of the conclusions from the histone transplant are that nucleosomes have evolved to be highly tuned to their own species chromatin interaction network, chromatin remodeling network. The linker region that separates the nucleosomes from each other is very long in mammals and very short in yeast, and it's determined by the host, not the histone. That's a very clear-cut result from this, which we really didn't know whether it would be the host or the histone, but it's a clear-cut case. And finally, we think that the reason they don't complement well is that they have a hard time shifting around when conditions change, and we have a number of instances, examples of that in the paper, showing that nucleosome positioning is largely correct, but when the nucleosomes need to move around in response to a change in the environment, they do so much more slowly than the native histones. Okay, so the last piece having to do with sort of gee whiz genomics we can do in yeast has to do with the species chromosome number paradox. So here's the tree of life drawn in a very beautiful and interesting way with today's organisms on the outside, the birth of the earth here at the center, and we have organisms that have values of n haploid numbers in the hundreds. We have organisms like the Australian jackjumper ant with n equals 1. Amazingly, in 1986, it was shown that the jackjumper ant has a single chromosome. And of course, being an ant, it's haplodiploid, so the males literally have 1 chromosome, and the females have 2. So that's the world record holder for the lowest. And then a not so distantly related butterfly is n equals 223. So we see tremendous diversity. Why is this? Interesting question. So with yeast, we have n equals 16. And we began a project to see, with our ability to manipulate the genome, can we get something closer to a jackjumper yeast? Okay? And so a student in my lab, Jingchuan Luo, came up with a plan for how we could fuse the chromosomes together by fusing telomeres and simultaneously deleting 1 centromere so as not to generate a dicentric chromosome. So the first step involved a series of these fusions to get to 4 roughly equal sized chromosomes, and then she made a plan to grow those into 2 larger chromosomes and perhaps ultimately 1 giant chromosome. I can tell you we're not there yet, but we're almost there. So these are the 16 yeast chromosomes here in the native strain, and what you can see is we've made a series here from n equals 16 to n equals 4 roughly equal sized chromosomes. The gel conditions used here started not to work very well for us anymore because everything bigger than this was running at the top of the gel. So we shifted to a different set of conditions. So now I'm showing you the 16 and the 4 strain. And now we can clearly see that we can make n equals 3, a couple of different versions of it, and even n equals 2. And we now have a couple of versions of n equals 2 as well. So amazingly, you can drastically change the haploid number of chromosomes. And really amazingly, the yeast doesn't care that much. Okay, so this is n equals 16 to n equals 4, a series of growth conditions, and I'm hard-pressed to see any difference in growth rate, including when we hammer the DNA with DNA damaging agents and Benamil to destabilize microtubules, various temperatures, and so on. With n equals 2, we start to see a little bit of a growth defect, but nothing at this level. So another example of how we can torture the genome in creative ways and get surprisingly fit organisms out. Okay, last thing I want to tell you about is how we're extending this to the study of mammalian genomes. In the background, you can hardly see it, but it doesn't matter, are all the GWAS hits mapped onto the human genome. And the basic idea here is, can we use this technology to understand the dark matter of mammalian genomes? So as you know, mammalian genes consist of tiny exons floating on a sea of large introns. And if we zoom out, we see that the distances between the genes are substantial. So it's a very different organization from the yeast genome, which is tightly packed like most microbial genomes. So where in all of that space are the control sequences? And of course, we have tons of fantastic genomic DNA data from ENCODE and ChIP-seq and so on and so forth that gives us lots of hints, but there's no good way to map these things to which gene they actually control. It's very challenging. And so we think we can potentially give a good input into this if we can rapidly assemble 100 kb genomic regions from mammalian genomes and then make designer modifications and combinations of designer modifications and then deliver those into the genome of, say, a stem cell, we might be able to learn a lot about which putative enhancer sequences are working on which genes and how they work together. So can we really do this? Well, Leslie Mitchell and others in our group have shown that we can. We've now made 2 approximately 100 kb mammalian loci. This is HPRT1 using a variant of swap-in that I described before, but it's an extra chromosomal swap-in. So here we have the HPRT gene and a 100 kb region containing it. And you can see that we can assemble this from amplicons we can make just by PCR, and then we could intersperse those with synthetic or semi-synthetic amplicons to make a lot of variants of that region. And basically, this was done in 3 steps in 1 month and worked the first time. And we think with some improvement, we can get this to 2 weeks to make a 100 kb locus once we have the starting materials in hand. So sometimes some of the PCRs are difficult, but the vast majority of them happen on a first try. Obviously, we have to work hard to deal with the repeats, but we happen to know a lot about repeats, so that helps us in this. And then we can recover these as backs from the yeast. And we have a very automated way to look through many yeast colonies for the winners that have the correct assembly. And basically that involves PCRing across all the homologous recombination junctions to find the winners. And that's what you see here on the lowest panel. Now the harder question is, how do you deliver this into cells? It's one thing to build it, but how do you deliver it? And what we've come up with is a custom landing pad for something we call Big In, for integrating big DNA molecules that, again, Leslie and her colleagues have worked out. And what you see here is we've delivered the HPRT into mouse embryonic stem cells, which have their own HPRT. And you can see the anti-human HPRT is picking this up. So despite the fact that it's been introduced into a mouse cell, it's being expressed at apparently normal levels in that mouse cell. And this works very well. Leslie was recently named a Stack Wunderkind on the strength of some of this work. Importantly, these various fragments have been of different sizes, have been integrated, and we have basically a phenotypic test for whether or not they went to the right place. And around 2.5% of the colonies in this experiment were the right thing, and PCR validation rates are extremely high, and sequencing is underway for the biggest one, but worked out only 1 base change in 50 kb for the first one. So this is the system that we've developed to automate the picking and evaluation of the yeast colonies. So this is a standard colony picker, liquid handler to make genomic DNA from the yeast colonies. Once the genomic DNA is made, The genomic DNA and the oligonucleotide primers and the master mix are combined into these 1536 real-time PCR plates, and what you're seeing there is the bulk reagent for the PCR being added. The primers are added, and the genomic DNA are added in this instrument here, which is an acoustic droplet ejection robot, which can basically create these tiny droplets of 2.5 nanoliters and fly them up into an inverted destination plate. So that's our 1536 plate turned upside down. And this enables us to assemble these 500 nanoliter reactions at a cost of 7.4 cents per genotyping reaction. And since the light cycler, the 1536 light cycler takes about an hour, we can run 30,000 of these reactions per day. So we can really, we really have the capability to make a lot of these custom genomic regions. So that's on YouTube if you're interested. So I'm here to tell you, we've built a lot of stuff, and we want to build a lot more stuff, and so operators are standing by with the order. And finally, we've had a lot of fun with this project. So we've married the multicolored yeast with the acoustic droplet ejection robots to make artwork. Okay, we started with a 384 biopixels here, and now we're in the 25,000 biopixel domain, so we can make very elaborate artworks. This one was made to commemorate the fact that we're very interested in engineering the genome of the pig. That's why we're talking to Recombinetics a lot. And we have a wonderful website about yeast art if you're interested in seeing more of the artworks. And with that, I will thank my wonderful lab and collaborators without whom none of this work could be done. Thank you very much.
Thank you very much. Do we have questions or comments?
So do you think it's possible to re-engineer to create a new mammalian genome?
Well, it's conceivable, it's possible. You may or may not know that George Church, Andrew Hessel, Nancy Kelly, and I have started an initiative called GP Right, which I didn't really get into, which aims to develop the technology to do just that. You know, there are major challenges in scaling up to an organism whose genome is 200 times larger and contains massively repeated regions, particularly in centromeres. For me, at this point in time, I think the sweet spot is the 100 kb to 1 megabase domain, where we can learn so much by really mining that. And so that's where I'm headed in my own lab for the next 10 years. But on the other hand, I've learned never to bet against George Church, you know? And I wouldn't be surprised if we can do that. I mean, there's already teams around the world that are talking about synthesizing entire human chromosomes. Whether that's the right thing to be doing today, I'm not sure. With the yeast project, we spent almost a year debating what that list of features should look like. What are the changes that are really going to pay off in the long run? You know, and some of them we haven't even begun to use yet, because you don't get to use them until the whole genome is finished. That was 10 years ago that we did that, and we still, you know, haven't really been able to exploit, for example, the stop codon swapping or the full intron deletion. That's like a massive delay of gratification. And also, it's really, a lot of thought has to be put into it, because obviously it's a massive investment, right? So I think we're going to see massive drops in the cost of DNA synthesis, and that will certainly help fuel the effort. But It's not going to happen tomorrow, but 10 years from now, it's going to be a really different world, and you all need to be getting ready for it. Yes?
With this type of technology, so you think the term speciation has become very big and trans species is possible?
Yeah, that's a really interesting question. So we had quite a, when we submitted the paper on the on these chromosome fusions, definitely I think we overreached a little bit on some of our claims in terms of maybe this was a new species, and that definitely got a lot of the reviewers very upset. But I think, yeah, I mean, the species concept has already been massively redefined based on the original biological species concept to today's sort of population phylogenetic species concept, and I'm sure the lines will be blurred further as we move along. I don't know, is our N equals 2, is that Saccharomyces cerevisiae? It's certainly not interfertile with Saccharomyces cerevisiae, and so by the biological species definition, it's not. Yeah.
So I have two completely separate questions. The first is, with your chromosome number experiment, have you subjected any of those lines to selective pressure? It seems that the larger chromosome number would be more responsive to selection.
Yeah, great point. So, you know, the yeast karyotype has, if you just look across the yeasts for values of N, all Saccharomyces are N equals 16. And then there's very strong evidence that there was a whole genome duplication. So there's a clade of yeasts that has a modal number of N equals 8. And the ones that have, say, N equals 6, you can show that there were 2 chromosome fusions, just like between, you know, chimpanzee and human, we have 1 chromosome fusion event. So, so my personal speculation on this, well, just to get to your question, there are also clade of yeast species with a modal number of 4. And they tend to be highly specialized species that grow in very specific environments, whereas Saccharomyces is a generalist. It's all over the place. And we know that you can get you know, changes in basically aneuploidies can arise at the drop of a hat in response to a selective pressure like what you're getting at. And that would be much more difficult to do if you're N equals 2. So we haven't done the experiments, but I strongly predict that they will be less able to adapt. But the reason this whole 4, 8, 16 thing is really fun is because it's gotten me thinking about the transition from prokaryotes to eukaryotes. If you think about it, you start with a single circular genome, it became a single linear genome, you might have a whole genome duplication to 2, then loss of genes and so on, and then a whole genome duplication to 4, 8, and 16. It's kind of a fun model. What's your second question?
The second one is your circular gene that you bashed and trashed and rebuilt. What was the phenotypic variation that you saw? Did you have more productive yeast?
So that was the right arm of chromosome 9. It's the shortest arm in the genome. It has, whatever, 43 genes on it. And what we did there, we knew that there were 2, we know a lot about the genes in the yeast genome, obviously. And there were 2 genes that were involved in amino acid biosynthesis. And so one was a lysine biosynthesis and one was methionine. So what we did was we plated out a whole bunch of cells and we just screened for things that either became lysine or methionine auxotrophs. So we knew they had undergone one mutational event. And other than that, there was no selection for anything except they had to be high fitness. They had to grow well. on non-selected media. So they were not selected for any gain of function or loss of function, other than those two auxotrophies. None of them are overtly better than wild type, although we haven't looked for like a really subtle effect. So other than those auxotrophies, we didn't really find much in terms of a phenotype. It was more, the purpose of the study was really to see what's the range of diversity at the structural level that we see when we just select for high fitness. And we were not expecting to see such a dramatic range of lengths, for example. The 3.7-fold longer has just massive duplications and quadruplications in it. We had one isolate that had like 13 copies of one region. It was really wild and unexpected.
Hi, excuse me. So I have a selfish observation. That being the chicken genome is 1/3 the size of the human, and it has a lot fewer repeats.
Yeah, I hear you.
And the other is, have you taken your scrambling system and asked the question, do you get any unique ones growing out under heat stress?
As a matter of fact, yes. So we've done some scrambling experiments. We've done what we call the Starbucks experiment, where we've scrambled strains and looked for resistance to caffeine and high temperature, and not at the same time. It turns out I didn't have time to go into this, but we've realized that there's one major downside to our original vision for scrambling, which was to do it in haploid cells, and that is anytime you get a recombination that pops out an essential gene, you're dead. So you lose a fraction of the population whenever you do scrambling in haploid. So now what we're doing is we're doing it in heterozygous diploids, where you have one set of unscramblable genes and one set of scramblable genes. So now if you lose a gene, the worst that can happen is you get a 50% drop in the copy number. But you can gain from the gain of functions and so on. And so it's in that context that we've been doing most of our scrambling experiments. And the interesting thing about it is you also can take advantage of the incredible diversity of yeast strains that are out there in the world. Yeast is used to ferment everything from chocolate to sake, you name it. So the sake yeast, and the reason I bring it up is it's intrinsically more temperature resistant than the other yeasts because of high temperature of fermentation. So we were able to get derivatives of sake yeast that were even more temperature resistant through one round of scrambling of these heterozygous diploids. So what does that have to do with chickens?
Well, I'm just interested in the global heat stress response. All learn from nature.
Yeah. Well, I think your point about avian genomes is excellent because it might be an outstanding stepping stone, between yeast and mammals. Instead of having to go 200-fold in scale, only 60-fold would be, or 65-fold would be a lot easier. So yeah, I'd love to talk more about it.
Quick follow-up, I just have to ask, the ones that the sake yeast, are they obligatory at a higher temperature, or are they?
No, but we did find with one of them was interesting. We got two really resistant derivatives that we're still trying to figure out the structure. One of them did better at high temperature and continued to grow normally at lower temperature, but one of them became very poor at growing at 30 degrees in normal temperature. So sometimes, when you select for performance under one condition, you lose robustness at other conditions. It's not surprising.
Any other questions? Okay, that was excellent. Thank you.
Thank you.